Well, it's a great pleasure to be here, especially to be introduced by Eric Fries. This is not my first time in Uppsala. I've been here, had the pleasure to have been here on a number of occasions, but none more pleasurable than this. I'd like to tell you that I feel very much at home. Now, this Uppsala cap has been signed by your Vice-Chancellor, but I would like to invite all of you to come down, only the students, afterwards, and I'd like as many of your signatures to take with me as a souvenir after this lecture. Is that okay? And I understand that after the lecture what you do is you throw the hat. So I am going to throw the hat after the lecture, not now, but I hope that someone will try to catch it and then come back up, and I would really appreciate some signatures so I can have a great souvenir for remembering this very lovely day. So the story, as you've heard, that threads, many of the threads through all three of our stories begin with the founder of modern cell biology, George Palade, whose work posed a number of questions, but none more central than the question outlined right here. He discovered that there is a trafficking pathway in the cell, especially involved in the secretion of proteins. We now understand, as Professor Sheckman illustrated, and largely through his work, the great breadth of this pathway, not just in evolution, but also the many avenues and branches of this pathway that operate throughout the cytoplasm. And we understand, as originally proposed by Palade, that this traffic is carried by vesicles. And I hope that you understood from the previous lectures that there must be many different types of vesicles in the cell, each of which carries a unique type of cargo going from a location where it originates, such as the endoplasmic reticulum, onto the Golgi, the pathway that Professor Sheckman has emphasized, or in many other locations in the cell. But the common principle involved, one way or another, is that each type of vesicle needs to deliver its cargo to the right place at the right time. If that fails, if that process of membrane fusion fails to either be sufficiently specific or on time, then cargo will be distributed incorrectly within the cell or even incorrectly in the body. Now, this delivery process, which we term vesicle fusion or membrane fusion, therefore, is fundamentally important for a wide variety of physiologically important and also medically important processes. Of course, there is the process of distribution of newly made proteins within the cell, which we've heard about in the last little while. And this, of course, is fundamental both for cell growth and for division. But also, many important proteins and other molecules are secreted from the cell entirely into the blood, whether it be the endocrine circulation or in exocrine physiology. And as a result, signaling molecules that integrate and produce integrative physiology throughout the body rely utterly on similar pathways. And then finally, as you heard in much more detail from Professor Suthoff, who has contributed so much to our understanding of the physiology and mechanisms of synaptic transmission, that similar vesicles, first spotted actually by George Palade, but first understood to be involved in synaptic transmission by Bernard Katz, are stored at nerve endings, ready to release their cargo, which in this case consists of many, many, many neurotransmitter molecules, to affect a potent neurotransmission event. All of these different biological and physiological processes, as I will emphasize today, operate according to one principle, the principle of membrane fusion. Now, this is actually a great surprise at some level. I mean, look at this diversity of physiology. One basic principle, which I'll emphasize, the SNARE complex. I remember one time going to a meeting in, I think it was in Constance, I'm not entirely sure. I got off the plane in the morning. In those days, I didn't fly business class, so I was quite dreary. And gave my talk, which at the time was not perhaps as sophisticated or molecularly advanced here. We hadn't yet discovered the SNARE complex. Thought it was an okay talk. Sat down. Next guy got up and gave a talk. I fell asleep. Then the next person, I won't mention the name of the person, got up and gave a talk. Woke me right up because he began by saying, well, Professor Rothman gave a very, very elegant presentation, as you just heard, but this has nothing whatever to do with neurosecretion. And it has nothing to do with hormone release, because he just told you that his process is sensitive to N-ethylamide, that it requires ATP, that it is inhibited by GTP gamma S, and we know that the release of hormones and the release of neurotransmitters behaves exactly the opposite. And therefore, there must be a fundamentally different mechanism involved. So that was the world in which we operated back then, because scientists sometimes look for complexity. They find it. Other scientists look for simplicity. They also find it. I've always fallen into the look for simplicity category, sometimes to a fault. Of course, the reason for this contradiction was that underlying both of these very extreme cases of physiology is a core mechanism, which can then be regulated differently. And if you look from the outside, from the point of view of physiology, you oftentimes have to work your way deeply into the core, and you get confused along the way. The wonderful thing about biochemistry and enzymology is that it tends to lead you directly to the core mechanism, and the reason is quite simply that when you make an extract and you dilute the components of the cell, only the most robust reactions can survive. As a result, the entire history of biochemistry has been successful in revealing core machinery, perhaps less successful in revealing machinery that adds a factor of 2 here or a factor of 4 there because of the dilution factor. That's a very fundamental point. Now, I came from a background in physics. All of my undergraduate studies were in physics. I always wanted to be a physicist. And even as a biologist, I've been trying to be a physicist. Now, that's actually hard to do, but nothing prevents you from continuing to think like a physicist. So from my point of view, the physics of membrane fusion is absolutely trivial. I mean, there are people that work on it, and I don't mean to insult you, because there's a lot more to learn. But at a certain level, it's trivial. Lots of membranes will fuse. And some of the simplest membranes are soap bubbles. They will also fuse. You know, a lipid bilayer membrane really is 2 soap bubbles that are concentric with each other. I don't think we're confused, nor have we been since the age of about 3, as to how soap bubbles fuse, and that they fuse spontaneously. If you hold them together long enough, they will fuse. And so really the problem of membrane fusion in the cell is no different, except that there has to be a precise regulation and control of this physical process to enable the right membrane to fuse with the right other membrane at the right time and at the right place. That's really the crux of the biology. But with this simple point of view, we began to attack the problem in the late 1970s, in a way by defining the problem first. And so I approached the problem by thinking about, trying to think, how would a physicist think about this? Now, physics, physicists always seek a universal explanation. So, and they always assume that whatever they discover will apply on every scale everywhere in the universe. If you think about it, that's a lot of hubris, right? I mean, you're Isaac Newton, right? The apple falls from the tree, okay? And you assume that you can explain the orbit of the moon around the Earth around the sun, and so on, from that, and that there would be a universal gravitational constant that you could measure that would apply everywhere. Now, any biologist that starts to think like that would immediately be put in the insane asylum, okay? But it doesn't mean we can't try. for some notion of universality. And you'll see, as you've seen in the secretory pathway, there is a certain element of that. So physicists always seek the simplest hypothesis that can explain as many of the facts as possible. Good. So looking at the problem that George Palade defined for us, which I first learned about when his work was quite fresh, when I was a first-year medical student at Harvard, and I was astonished to see the kinds of electron micrographs that he captured, that Randy showed earlier, as a matter of fact, where, and this, by the way, is not his only micrograph. It has been said that he has actually produced other micrographs. But anyway, this is the one we all show because it is so beautiful. And it illustrates the rough endoplasmic reticulum, the budding of the vesicles. And when you look and appreciate this from another level, look how beautifully organized it is, right? I mean, here you have the endoplasmic reticulum right up against the entry into the Golgi apparatus, literally stuffing vesicles into the mouth of the Golgi apparatus. It's as if the physical proximity of the two were absolutely necessary. And that, in fact, was the prevailing wisdom at the time. When I was starting at Stanford as an assistant professor, I went to several of the popes in the field at the time, and I explained to them that I would like to try to reproduce all of this in a cell-free extract. And they said, by the way, you really shouldn't bother because it is the anatomy that dictates the specificity. The physical apposition of the organelle that gives rise to the vesicle onto the organelle that receives it literally hands the vesicle and stuffs it in the mouth of the recipient membrane. Therefore, if you were to disperse these organelles in the cytoplasm, you would only create chaos and probably see nothing. On the other hand, I never believed that for 1 minute. And that's because I came from this background in physics. And rather, I thought the simplest idea, and this is something we published very early on in the work before any of the molecules were known, was that there had to be a cycle that would operate in which there would be intrinsic chemical specificity encoded in the surface of the vesicles to determine where they would bond and where they would fuse. And if that specificity was potent enough, it would actually, when allowed to operate in a cell, give rise to this morphology and anatomical proximity as the very nature of the outcome of the process. Not the cause of the process, but the outcome of the process. So that really was our fundamental hypothesis, that the intrinsic chemical specificity governs transport, not anatomy. And that gives rise to a remarkable prediction, which is that vesicle traffic, which, as you've heard, generates the very anatomy of membranes in the cell, nonetheless can take place in the absence of any pre-existing organization in a cell-free extract. So that was our underlying hypothesis that we wanted to test. Well, the next thing you need, once you have a hypothesis, is someone who shares your insanity. That was Eric Fries. Now, Eric joined the lab in 1978. In fact, he was the first person to join my young laboratory at Stanford. He just arrived, having recently, I should have to say, discovered that viral envelope fusion occurs inside the cell, a critical observation that led to the discovery of the endosome, as well as a number of other important discoveries. And Eric was the brave soul who undertook with this homogenizer to reproduce for the first time vesicular trafficking in a cell-free extract. I gave this token to Eric. It says more Dounce per ounce. This is a Dounce homogenizer. That's why we said that. This was a going-away gift. And Eric has kept it all these years. And just a couple of days ago in Stockholm, donated it to the Nobel Museum at their request as a kind of an artifact. How does it feel now to be finally generating artifacts after a career of successful experiments here in Uppsala, having done many fine things since leaving my laboratory? Well, anyway, Eric, I will always owe you a huge I will always have a huge debt of gratitude. And what Eric and I designed was a method, and you'll find this in all the textbooks. I won't take the time. Time is running short to describe it. But we worked out a method where we could incubate extracts from two cells, one of which would provide a cargo protein, and the other would modify the cargo protein, but only if the cargo protein could move between the two cell extracts. And the elegance of that was there could be no pre-existing cellular organization between extracts from two separate cells, right? Okay. Well, we thought right, but the reviewers of the paper, they weren't so sure. But it did eventually get published, and we were following this work, and there was a lot of work that went into this, because it worked, and then it didn't work, then it worked, and it didn't work. And Eric will remember how he sorted through the fact that it didn't work, and the reason it didn't work is we thought we reconstituted ER to Golgi transport, but in fact, it turned out, as Eric found, that it was really a transport process within the Golgi that was reconstituted, which was my personal nightmare. It was fine for him because he left, okay? But for me, it was an absolute nightmare because how would you like to reconstitute a process that nobody knows existed at the very same time that you're claiming to do something that everybody said was impossible? Well, that was the next 10 years of my life. And as Randy indicated, when we found that his genes and my proteins that came from this assay united, I think the field as a whole felt grateful, and I know that that the geneticists finally felt a sense of relief now that the genetics was validated by the biochemistry. So out of this initial effort came a faint little band, and this was our first successful experiment in 1979, followed by, as I've mentioned, many unsuccessful experiments. But success returned eventually again. When Bill Balch was in my lab as a postdoctoral fellow, he and I worked very closely in the lab to modify this assay and make it more workable for enzyme fractionation. Instead of a little band, we had a big signal because we were directly measuring the incorporation of the sugar rather than the shift of a small band on a STS gel. And you know, we found that it required a high-speed supernatant because there are proteins needed to make the vesicle, which eventually we found, and other proteins needed to fuse the vesicle, which we also found. And I will now turn the remaining minutes to how we found those and focus on the process of membrane fusion. And that came about through this inhibitor N-ethylmaleimide because when this, we added this inhibitor, you see the amount of transport goes way down. The reason it goes way down is because vesicle fusion has come to a stop. The vesicles continue to bud, and in collaboration with Lelio Orci, and I have to take the credit actually for having introduced Randy to Lelio, so that he too, as I, could benefit from his profound impact on this field. I regret that Lelio Orci's name wasn't mentioned in the Nobel Prize Committee's summary of the field. There were a number of other contributors to the field, of course, apart from the three of us who contributed one or the other stage to, quite importantly, to our present understanding. And unfortunately, the rules of the Nobel Prize limit the number of recipients to 3. But especially Lelio Orci contributed by uniting biochemistry and genetics with electron microscopy to provide orthogonal methods that allowed each of us in our own way to separate the stages. And in my case, it was done by using an inhibitor rather than a mutant. But in concept, a specific inhibitor causes a chemical lesion, which in the end is similar to a genetic mutation. And what it's unfortunate with the lights up, you can't see it too well, but these are vesicles that formed, and we showed in parallel experiments they formed with what are now called COP1 coats, and that these COP1 coats were removed when the GTP was hydrolyzed, similar to the process you heard about from Randy with COP2 coats. And now the vesicles were left to fuse, but they could not fuse. And this led Vivek Malhotra and Lelio and myself to conclude in the late 1980s that there must be an N-ethylmaleimide sensitive factor, which we called NSF. Sorry, the inevitable cold that you develop, not from the fair weather that you've had this time of the year, but from the large accumulation of biological weapons that are concentrated in Stockholm from around the world at this time. You know, ironically, I think I caught a cold at the Nobel Foundation yesterday from the gentleman representing the organization who was banning biochemical weapons and trying to harvest them Apparently, he harvested them from all over the world and showed them off when he came to Stockholm. And Randy and I and Tom, and in fact, one of the economics laureates absolutely almost keeled over. Although it was economics, so there wasn't much missed, you know, so on. Anyway, so we then purified this N-ethyl-malayamide sensitive factor based on its ability to restore the assay signal to this NEM inhibited reaction. So what we have is a protein then that's pure. We discovered it was an ATP hydrolyzing enzyme. It's a cytosolic protein, and it's needed, as Randy indicated, it's the same as Sec18. NSF and Sec18 are needed for fusion throughout the cell. So somehow its hydrolysis of ATP is being used in one or another way, directly or indirectly, to power membrane fusion. But it can't be providing specificity, right? Because it's being used everywhere, 10 different types of vesicles or whatever, each fusing to 10 places, can't use the same enzyme, can use it as an engine, but not to provide specificity, right? Just common sense. Alpha SNAP Sec17. We also purified that because it was needed to bind NSF to the membrane. Also turned out to be a universal component of the fusion machinery. Engine, not specificity. Also water soluble, which is great because it can be provided everywhere throughout the cell equally. But somewhere there has to be something in the membrane. That turned out to be the receptor for the SNAP protein, which we call SNARE. SNAP receptor, S-N-A-R-E. That's where the word SNARE comes from, SNAP receptor. And we noticed that SNAP protein binds to membranes like the Golgi membrane in a saturable fashion. There must be a SNAP receptor. But NSF does not. NSF only binds and hydrolyzes ATP after it has bound to SNAP. And the primary localization to the membrane is the binding of SNAP to the SNAP receptor. So the next stage in the project, and the pivotal stage, became, could we identify the SNARE proteins, the SNAP receptors? Okay, if we did, we would likely then have membrane proteins that might be more directly involved in the fusion process. And as we knew that membrane proteins, there are usually different membrane proteins in each different compartments, we imagined there would be a set of SNARE proteins in the ER, another one in the Golgi, another one in the plasma membrane, and so on. They would probably be related to each other, but they wouldn't be the same, and their interactions with each other could provide the chemical specificity that we had predicted so many years before. Well, at this point, along came another brilliant scientist, Thomas Zollner. He just arrived from Munich, having identified any number of important proteins in the mitochondrial outer membrane in his work with Walter Neupert. And he brought a level of biochemical skill to the lab that we sorely needed at the time, along with a German-speaking contingent, I might add. As a result of his coming in, half of Walter Neupert's lab came then in the few months afterwards. Everyone in the lab spoke German, and I began to learn Gym English. And I won't go through the details of the method that Thomas used, but essentially he used the inherent properties of this ATP hydrolysis cycle to fetch the receptor out of the membrane and then to release it from the SNARE, with ATP hydrolysis in two successive steps of affinity purification. He also, almost by accident, used brain, cow brain, as a source of the membranes. This is actually quite important historically. Not that cows are important historically. I mean, I don't want to offend anybody who loves cows. There may be people here who love cows. But it's important because we were working, Randy was working with yeast. We were working with, tissue culture cells, some more or less generalized fibroblasts, mostly CHO cells. We were certainly not intending to work on synaptic transmission. We measured, however, the level of the SNAP receptor, the amount of binding capacity per gram of protein, and it was 10 times higher in the brain than any place else. Now, if we'd have had any brains at all, we'd have understood that that's because the brain is the most active organ in the body for fusion because of all those synaptic vesicles that you heard about from Tom. But we weren't that smart. We were just dumb biochemists. And so we used brain extract. And out of that, therefore, as a result, we identified the SNARE complex, that is to say, the SNARE proteins that are involved directly in membrane fusion. We discovered those in the context of the brain, which means, by the numbers, in the context of synaptic transmission. There are many SNAREs in the brain because brain cells, neurons are, after all, cells. But they are highly specialized, and therefore the bulk of the SNAREs in them, and therefore the bulk in the brain, are VAMP, syntaxin, and SNAP25. VAMP, or synaptobrevin, syntaxin, and SNAP25. So when Thomas Solner extracted the SNAREs, he found, to our great surprise, a complex of proteins. All three of them were known at the time, but it wasn't really known very much what they did. Okay? They were known in part because of the work of Tom and a number of other colleagues at other universities who systematically were studying and elucidating the molecular components of the synaptic vesicle, Tom and most especially Richard Scheller. But there were others. Syntaxin was known from the work of Akagawa and Scheller, and it was known, separate work, to be present in the plasma membrane. But it wasn't known exactly what it did, but it somehow was thought to be involved in exocytosis, directly or indirectly. SNAP25, synaptosome-associated protein 25 kilodalton, was known from Michael Wilson's work. It had been cloned. It was known to be in the synapse somewhere, but it wasn't even thought to be in association with these proteins, never mind actually on a synaptic vesicle or the plasma membrane. But finding this complex, bridging the vesicle to the membrane, and in the context of the general components that power the fusion machinery, NSFs, ATPase, and SNAP, immediately led us to grasp that the SNARE complex might be the very general principle that we were looking for all along that could encode chemical specificity and at the same time power membrane fusion. And so we immediately, and somewhat boldly I might say, generalized this into a hypothesis which has proved to be largely correct, in which we imagined that there are homologs we called these SNARE proteins, SNAP receptors, all of them, and we called this the V-SNARE, and we called that the T-SNARE. V-SNARE for vesicle, T-SNARE for target membrane. And we imagined that just like in this synaptic vesicle situation, the other vesicles in the cell, the many vesicles that Randy had studied and his students had elucidated so beautifully, and which George Pilati had first seen in the electron microscope, the many types of vesicles, each would be provided with its own version of the V-SNARE, its own version of VAMP and synaptobrevin, each of the target membranes with its own version of the T-SNARE, a structural equivalent to syntaxin and SNAP25. And we further envisioned that each V-SNARE would be designed specifically to form a cognate match. It would specifically pair. So V1 would bind T1, V2 would bind T2, V3 would bind T3. And that type of specificity, combined with somehow the ability of the SNARE proteins to engage in fusion, would both at the same time would allow delivery at the right place and at the right time. Now, there's a lot of evidence that's accumulated in favor of this hypothesis over many years, which make it very clear that it affords an essential level of specificity for membrane fusion. It is not by any means the end of the story. We heard about Rab proteins. You probably heard, I'm assuming in Tom's lecture, about Rim proteins. And there are many other proteins that are involved in adding layers of specificity and regulation to this process. But at the crux, there is an inherent specificity that is very powerful in the SNARE proteins. And there's no experiments that show this better than these next two experiments, which I'll mention very briefly. In 1998, we found that you could take the synaptic vesicle SNARE proteins out of the synapse, simply make them as recombinant proteins, and provide them to artificial bilayer vesicles, the V-SNARE in one vesicle, the T-SNARE in the other, no other proteins, and they will fuse. And they will fuse automatically, and they will fuse extremely efficiently. They fuse, and I'll come to this in closing in a moment, much more slowly than the millisecond synaptic release, orders of magnitude more slowly. That is a difference that we are only now coming to understand in structural detail. But the fact is they fuse with great potency. What I find personally the most satisfying result, again, from a physical chemical point of view, is shown here. This represents probably 20 person-years of work in this one slide. I mention only one name because James McNew was the lead author of 3 papers that appeared in Nature in the same issue in the year 2000. This is a trifecta that is rarely accomplished today. I tried the same trifecta with Randy's new journal that no doubt you've heard about on other occasions, eLife, and I was successful in achieving 2 publications, not 3, but we'll be back. No doubt. No doubt. Now, what we did here was we took advantage of Randy's, the level of understanding of the genetics of the secretory pathway that by then had arisen and was very complete. And we expressed every single SNARE protein encoded in the genome of yeast. So we had a large number of proteins. Each of these T-SNARE proteins is actually 3 separate proteins, typically. So there is a very large number of proteins. And we purified each one, and we then put them into liposomes as pure proteins, 1 at a time, 2 at a time, 3 at a time, 4 at a time, 2 over here, 2 over there, 3 over here, 1 over there. Can you imagine all of the combinations that we did? And out of those hundreds of combinations, the only ones that fused the liposomes, and did so very efficiently, are the handful that are shown here. None of the others, and these are only a small number of sampling of all the other combinations, gave any fusion at all. And these ones correspond to exactly the steps that Randy had discovered for transits between the compartments in the yeast cell. Exactly. Now, what does that mean? It doesn't mean we could replace Schechtman. He is irreplaceable, as you no doubt understood from his lecture. But what it does mean, I say that with great friendliness, of course. What it does mean, okay, is that if you had a physical chemist and if you handed that person just the genes, and that person did this experiment, they could then go back to the yeast cell with no additional knowledge and say, here are the vesicle trafficking pathways that could occur in the cell. And that physical chemist would predict exactly what they would be with no knowledge other than the inherent physical chemistry of these proteins and the resulting property for fusion or not. And that the results of that physical chemical experiment would lead to exactly the pathways that Randy and his colleagues discovered. That is how powerful this is as a predictor. So finally, how does the fusion process actually occur? Well, it requires, of course, the assembly of the SNARE complex, because the SNARE complex by itself can promote fusion. Reinhard Jahn and his colleagues made an important discovery in the same year, actually, that we described the fusion by SNARE proteins. And he and his colleagues, and most especially he and Axel Brunger, the crystallographer, discovered that the SNARE complex consists of a 4-helix bundle. And the V-SNARE contributes 1 helix, the T-SNARE contributes 3 helices. This has turned out to be true over and over and over again. And they proposed a model which has turned out to be essentially correct, that this SNARE complex assembles between the membranes in a polarized fashion, from the membrane distant N-termini towards the membrane, a process they called zippering. This zippering, it turns out, occurs in 2 discrete stages. First, the bilayer, first half the bundle zippers. This came from later work from several laboratories. And then the rest of the bundle zippers. That causes a natural pause point, which I'll illustrate to you, halfway through the zippering. That pause point is used for regulation. It's designed to be there. It actually is how Tom's vesicles get released so efficiently and quickly, because the synaptic vesicles have lined up and they've been forced to pause halfway through the zippering process, and that pause is essentially made permanent. Permanent until synaptotagmin has bound calcium, and then that block is one or another way removed. We're all trying to figure out exactly how that works. We don't know exactly yet at a structural level, but that's sort of where the field is. Now, in the process of this zippering, there is an inner-directed force that is produced that literally, and it is the force due to protein folding. The SNARES, to a good first approximation, are unfolded proteins. When they fold and assemble into this 4-helix bundle, they assemble into a structure that is rock solid. You almost have to boil the water to separate the SNARES, okay? The energy that is released from that spontaneous process of protein folding, that is channeled into the membrane, and it is that forces the 2 soap bubbles together, okay? Of course, that means that the SNARE complex, to be used again, has to be busted up there's two choices. You either can heat the cell to 90 degrees, not recommended for biologists, or you can use NSF, which hydrolyzes ATP, linking to the SNARE complex only after it assembles, with SNAP as an intermediary to literally wrench it apart, essentially creating thermodynamically a high temperature locally, unfolding the proteins so they can now go back to another cycle. That is the way in which NSF is the engine, so to speak, that allows recycling. The engine that drives the fusion process is the energy released from protein folding as these proteins go downhill. You'll see this more vividly here. We start out with an unfolded V-SNARE and a largely folded T-SNARE, although there is a lot of biological complexity in the folding and assembly of that T-SNARE. And there are processes, especially in the synapse, that require the MUNC18 and the MUNC13 genes and some of the other proteins that Tom has described that are upstream and lead in a regulated way to its assembly. But once it's assembled, the V-SNARE starts zippering in. It zippers to the halfway point. That's where it pauses, essentially, for regulated exocytosis in the case where you need to have millisecond release, because it only takes less than a millisecond for the rest of the zippering to occur. As zippering goes to completion, the bilayers are brought very close to each other. This puts the water in the middle, so to speak, under a lot of pressure. My colleagues and I have made estimates, they're rough estimates, of what the pressure is in here. And at the moment when it is most pressured, we estimate it is between 100 to 1,000 times the pressure of the atmosphere. At normal atmospheric pressure here, 1,000 times atmospheric pressure here. Now, lipid bilayers are well understood to exit from the bilayer structure into non-bilayer structures, most especially micelles, at pressures above tens of atmospheres, depending on the lipid composition, but certainly by 100 atmospheres. And we are quite sure that that's why the soap bubbles form or fuse, because the layers of these soap bubbles are eliminated as planar surfaces under the hydrostatic pressure. There is no proof of this model. If there will ever be a proof of this or any other model of coalescence, it will come essentially through molecular dynamic simulation using methods similar to that this year's chemistry laureates have developed for small molecules. In any case, one layer after the next is eliminated, and then the force that produces this pressure is eliminated and vanishes as the snares are fully zippered. And now the V and the T snare are in the same membrane, and in fact they zipper even within the bilayer right the way through to incorporate the vesicle into the target membrane. Now, to summarize, in case anyone here missed my point, what we have is an underlying mechanism which is, yes, at some level complex, but at another level no different than a lady's hairpin, okay? Like a hairpin, and I don't mean to offend gentlemen who, by the way, may wear hairpins. Like a hairpin, it takes force to pull the hairpin apart. The stretched hairpin is like the unfolded snare pin. Snare pin is my term for the pin that's used for biological fusion, because the snare complex forms a pin when it pins the two membranes together. Now, there's an important difference. In biology, a snare pin forms from two parts. One part comes with one vesicle, and the other part of the pin, the other half pin, so to speak, the T or the V parts, they come each with a vesicle. They have to link to each other to bring the one vesicle to its target membrane. That's where the specificity comes in. Where the force comes in is the folding energy, which is corresponding to the assembly of this pin. Now the pins have bound each other, and V and T assemble, zippering in just like this pin, forcing the two vesicles together. Now, I think you can imagine, and I don't really know how to stop this video, I'll let it run one last time, that if the surface tension of these two balls is exceeded by the force that's pushing them together, it's just simple physics, what's going to happen? They'll coalesce. But if the pin does not exert enough force to overcome the surface tension, they will merely bump into each other. So two hard balls will stay like so. But two soft balls, and that's what membranes are, deformable balls, will inevitably be fused. And so the elegance and the beauty of this is that nature has provided the cell a dozen or so half V pins and a dozen or so half T pins. And they come together at the right place at the right time. They assemble to form the physical equivalent of a hairpin. And as they do that, they overcome the surface tension to force these two soap bubbles together. Now, I've run out of time, so I won't show you the experiments that demonstrate this actually with single molecules that we can pull apart, nor will I show you our recent crystal structure, see, I just showed it to you, that in which the complexin molecule can be seen sticking its finger into the last part of the SNARE complex, preventing this last half from zippering, preventing exocytosis at the last millisecond. This is certainly not the whole answer to what produces the clamp and which allows the synchronous release mediated so importantly by the very fundamental mechanism that Tom has discovered with calcium and synaptotagmin, but it will be an important element of that process. In many laboratories, I know my laboratory, I suspect Tom's and probably others, are working hard to try to understand the atomic level details of how this works. We understand the outlines of it, the physiology is clear, and it remains to us and maybe you, hopefully, to teach us more about how this works. So I will just simply conclude by thanking the many dozens of contributors. I don't have pictures today of the hundreds of people that have worked in the lab, but I just, with one slide, like to thank the many dozens of people who've helped me in a lifelong study of bubbles. This is a picture, actually, if anybody's interested, I can tell you about it, taken by a Japanese photo artist. It has been exhibited in various places, and she was nice enough to give me a copy, and she asked me, you should do something that represents your science and also who you are. And so I thought, she thought I was going to write out some equations or something at the board, and I thought, gee, what could be more fun than bubbles? Blowing bubbles, childlike attitude. I like to think I've never grown up. Now with the Nobel Prize, it's a little hard to continue to feel that way. Also, having grandchildren doesn't help either. But in any case, the bubble is beautiful. A bubble is symmetrical. A bubble is what we study. We blow bubbles. We consume bubbles. This year with The Economist, they also work with bubbles. The difference is that, as I tried to explain to The Economist, that they fight about whether bubbles exist or not. Now, the beautiful thing about science is, we can have a discussion about who discovered what bubble first, okay? But we don't debate that there are bubbles and that they're of central importance to all of life. And so thank you all very much. And I hope that someone will catch this. Please bring it back.